Have you ever wondered how DNA forensic science works? How detectives solve crimes by analyzing just a tiny bit of biological evidence? Welcome to the intriguing world of forensic DNA science, a field that has revolutionized criminal investigations and the justice system as we know it. The power of this science lies in the unique genetic blueprint that each of us carries within our cells, our DNA. DNA, or deoxyribonucleic acid, is essentially a biological instruction manual that makes you, you. Every cell in your body carries a copy of this manual, and it's this ubiquity that makes DNA such a powerful tool in forensics. Whether it's a strand of hair, a drop of blood, or a swab from a coffee cup, these can all provide the crucial DNA evidence needed to solve a crime or identify an individual. Forensic DNA science involves a range of techniques to analyze these tiny samples of DNA. These methods can reveal an individual's genetic profile, akin to a biological fingerprint, and this profile can then be compared to others to determine relationships or identify individuals. This is a process often referred to as DNA fingerprinting, but don't be fooled by the simplicity of the concept. The process of extracting, analyzing, and interpreting DNA evidence is a complex one. It requires meticulous attention to detail, a deep understanding of genetics, and a keen awareness of the legal and ethical implications of such analyses. DNA evidence has become a cornerstone of modern criminal investigations. It provides an objective, scientific means of identifying or excluding individuals as suspects, helping to deliver justice. Its importance cannot be overstated. It has led to countless convictions, exonerations of the innocent, and has even helped to solve cold cases that have remained unsolved for decades. By the end of this video, you'll have a basic understanding of how DNA forensic science works and the key terms associated with it. We'll delve deeper into the techniques, terms, and statistics behind this fascinating science. So, stay tuned as we unravel the complex yet captivating world of forensic DNA science. To delve deeper into the subject, we need to understand some key terms used in DNA forensic science. Let's start with STR, which stands for short tandem repeats. These are small sequences of DNA that repeat themselves in a row on the DNA strand. They're like the unique patterns in a barcode, and they're highly important in forensics because they can help identify individuals. Next, we have VNTR, or variable number tandem repeats. Similar to STRs, these are also repeating sequences of DNA. However, VNTRs are noticeably longer. They're essentially the big brother of STRs, and their variability is what makes them useful in DNA profiling. Moving on to RFLP, the restriction fragment length polymorphism. This technique involves cutting the DNA at specific points and comparing the lengths of the resulting fragments. It's like slicing a loaf of bread and comparing the sizes of the slices. The RFLP method was a pioneer in DNA fingerprinting, but it's less commonly used today due to the advent of newer techniques. Now let's talk about PCR, the polymerase chain reaction. This is a method used to amplify a specific region of DNA, making millions of copies from a tiny sample. It's like a photocopier for DNA, and it's crucial in forensics because it allows scientists to work with small or degraded samples. Next up is SNP, or single nucleotide polymorphism. These are variations at a single position in a DNA sequence among individuals. Think of them as typos in the DNA script. SNPs can help us understand the relationship between genetics and disease, and can also be used in individual identification. Finally, we have mtDNA, short for mitochondrial DNA. This is a small circular piece of DNA that's inherited from your mother. It's a superstar in forensics because it can be used when nuclear DNA is scarce or degraded, such as in hair shafts or old bones. These terms may seem a bit overwhelming, but they are the building blocks of forensic DNA science. One of the most fascinating aspects of DNA forensic science is DNA fingerprinting. Quite the marvel, isn't it? DNA fingerprinting is the process of using fragments of DNA to create a unique fingerprint or profile for an individual. Let's dive into how this works. It all starts with a DNA sample, which could be from blood, saliva, or hair, for instance. This DNA is then broken down into smaller fragments using enzymes. These fragments are then separated based on their size using a technique called gel electrophoresis. The result? A unique pattern of bands that represents the DNA fingerprint. But how do we read these fingerprints? This is where the terms we discussed earlier come into play. Short tandem repeats, or STRs, and variable number tandem repeats, or VNTRs, refer to sequences in the DNA that repeat a specific number of times. These repeats vary from person to person, making them essential in creating a unique DNA fingerprint. To further enhance the accuracy of DNA fingerprinting, we use techniques such as restriction fragment length polymorphism, or RFLP, and polymerase chain reaction, or PCR. These techniques help us to cut and replicate the DNA, creating more fragments and thus a more detailed fingerprint. DNA fingerprinting has a broad spectrum of applications in forensic science. It can be used to identify suspects in a crime, determine parentage in paternity cases, and even identify bodies in disaster situations. It's a powerful tool in the hands of forensic scientists, offering a high degree of certainty when it comes to individual identification. Remember those single nucleotide polymorphisms, or SNPs, in mitochondrial DNA or mtDNA we talked about? They too play a crucial role in DNA fingerprinting, providing additional layers of information that can further refine the fingerprint and make it even more unique. DNA fingerprinting is like a unique signature that can pinpoint an individual amidst millions. It's a testament to the incredible power of DNA and the intricate science behind it, using the very building blocks of life to solve mysteries and seek truth. The interpretation of DNA forensic statistics is another crucial aspect of this science. It's like unraveling a complex mystery. Each piece of DNA evidence carries with it a trove of statistical data. These statistics can give us a deeper understanding of the likelihood that a particular DNA sample comes from a specific individual. To interpret these statistics, we often use probability calculations. Imagine you have a DNA sample from a crime scene and you found a match with a suspect. You might wonder, what are the chances that this match is just a coincidence? Let's walk through a hypothetical scenario. Suppose that the chance of a random person matching this DNA profile is 1 in 10 million. In a country with a population of 300 million, we would expect about 30 people to have this DNA profile by chance. But, before you start rounding up those 30 people, remember that this is a simplified example. In real-world forensics, the calculations can be much more complex, taking into account factors such as related individuals, population subgroups, and the possibility of laboratory errors. These complexities remind us that while DNA evidence can be incredibly powerful, it's not infallible. The statistics are only as good as the data and assumptions they're based on. That's why it's so important for forensic scientists to be meticulous in their work, from the collection and analysis of samples, to the interpretation of the results. And remember, forensic DNA statistics don't operate in a vacuum. They're just one piece of the puzzle in a criminal investigation. They need to be considered alongside other evidence, from eyewitness accounts to fingerprints. In the end, the interpretation of DNA forensic statistics is not just about crunching numbers. It's about using those numbers to tell a story. A story of what might have happened, who might have been involved, and how certain we can be about these conclusions. Interpreting these statistics accurately can make the difference between guilt and innocence. The final piece of the puzzle in DNA forensic science is the DNA database. Imagine a vast virtual library where each book represents a unique individual's DNA profile. That's essentially what a DNA database is. It's a collection of DNA profiles stored digitally, ready for comparisons and matches. Now, how are these libraries formed? Well, it all starts with collecting DNA samples, either from crime scenes, suspects, or even from certain categories of convicted criminals. Once these samples are collected, they're processed in a lab where their individual DNA profiles are established. These profiles are then added to the database, forming a new volume in our virtual library. The beauty of these databases lies in their ability to aid investigations. Let's say investigators find an unknown DNA sample at a crime scene. They can run this sample against the profiles in the database in hopes of finding a match. If a match is found, it could lead directly to a suspect or provide other valuable leads. However, these databases aren't without complexity. There are ethical concerns around privacy and consent, as well as the potential for misuse or misinterpretation of data. It's a delicate balance between utilizing technology for justice and upholding individual rights. Furthermore, the databases themselves are complex. They need to be constantly updated and maintained for accuracy, and the sheer volume of data can be overwhelming. It's not just a matter of storing information, but also of finding efficient ways to search, compare, and analyze this data. Yet despite these challenges, the importance of DNA databases in forensic science is undeniable. They're an invaluable tool in the fight against crime, providing a way to link suspects to crime scenes, identify repeat offenders, and exonerate the innocent. DNA forensic science is a vast and complex field, but understanding its basics can give us a glimpse into how science aids in solving crimes and delivering justice.